We will now talk about the genetics of axis specification in Drosophila. As you can see from this organizational chart, Drosophila is, of course, an insect. It has an exoskeleton, but it also has jointed appendages along with several other groups of arthropods. Unlike the nematode, C. elegans, which we studied a little bit earlier, this then has a little different mode of transportation in terms of having wings and legs to be able to get around. As a result of studies from Thomas Hunt Morgan's laboratory during the first 2 decades of the 20th century, science knows more about the genetics of Drosophila melanogaster than that of any other multicellular organism. As a result of these studies, Thomas Hunt Morgan won a Nobel Prize in Medicine or Physiology in 1933. This work was for showing that chromosomes carried the hereditary information. His work on Drosophila, then, really has formed the basis of modern science, the modern science of genetics. Drosophila has several advantages as an animal model for laboratory research. Drosophila is easy to breed, hardy, prolific, tolerant of diverse conditions, can live in a very small environment, is very inexpensive to rear, and it has a number of other advantages as well, primarily now based on the extreme number of genetic mutations that have been discovered in Drosophila, and many of those mutations relate directly to some of the higher vertebrates, including humans. Drosophila, however, as a developmental biology tool, wasn't perhaps as good as some of the other organisms that came along, in particular because the fly embryos proved complex and intractable, being rather difficult to see through when you looked in a microscope. They're non-transparent, in other words, not really large enough to manipulate experimentally like some of the amphibian embryos, and it was really not until the techniques of molecular biology allowed researchers to identify and manipulate the insect's genes and take advantage of all those many mutations and the RNA differences that its genetics could provide, that it really became an important tool to understand developmental biology concepts. And when that happened, the revolution occurred in the field of biology, and particularly developmental biology. And this is further advanced now since the early 2000s, when the Drosophila genome was totally sequenced and the ability to generate transgenic flies at a high frequency became routine. Researchers are now able to identify developmental interactions taking place in a very small region of the embryo and to identify enhancers and other regions of the genome that control developmental processes. The merging of our knowledge of the molecular aspects of Drosophila genetics with our knowledge of the fly's development has built foundations on which the current sciences of developmental genetics and evolutionary developmental biology, in fact, are based. If we look at early developmental biology, in the early Drosophila development, the cell membranes do not form until really about division 13. And in the early stages, the nuclei divide, but they don't really form into individual cells. To orient you on this particular slide, top part of the embryo is the anterior part, the bottom part the posterior part, just for orientation. Now, if you look through the sequence of numbers, this is indicative of the number of cell divisions that have taken place. The first division, number 1, second division, and so forth. And as you go through from 1 to 9 or 10, you're seeing most all of the nuclei are in the center part of this huge ovum, this zygote at this point, but this huge zygote which has a large number of syncytial nuclei within it. So it's really still, even at the 9, 10, and 11 stages, it's still a single cell with numerous nuclei, which by the 11th and 12th divisions, those nuclei line up along the periphery of the cell, and by the 13th, 14th cell divisions, those cells start to incorporate the membrane from the ovum and become individual cells. Also, during those stages, you get the formation of germ cells. Meiotic divisions are taking place, and down here you can see that there are germ cells that have begun to form at that point in the ovum. As I said, even though nuclear division is taking place, cells have not really formed, although each of these nuclei is encircled by sort of a microenvironment that allows cells to form very quickly once these nuclei come in contact with a membrane on the outside of the developing embryo. Looking at nuclear and cell division in a little more detail then, if we look at panel A, what we see is that the nuclear division, but not cell division, has taken place. So this is another better look at what this syncytium looks like. You can see that the individual nuclei are actually in the process of anaphase at this point. The chromosomes have formed, and each is surrounded by sort of a microenvironment that's like a cell, except that the membrane hasn't formed. And at higher magnification, you can see that these cells also are formed. The chromosomes are dividing at the cortex of a syncytial blastoderm, although there are no cell boundaries. Actin, which is green, can be seen forming around the peripheries of the cells, and also you see a lighter color that indicates the presence of sequestered cytoplasm in those areas, but not sequestered within a membrane. In C and D, here you see in C, these are interphase nuclei that have now moved to the periphery of the cell. It's starting to be surrounded by the membrane of the ovum, and here you're seeing cells dividing parallel to that membrane. These cells are in anaphase, and they're going to divide. They won't yet be cells, but rather just nuclei, although they function more or less like cells because of the microenvironment that surrounds them. If we look at the formation of the cellular blastoderm in Drosophila, the nuclei divide within a common cytoplasm, as mentioned before. However, each nucleus within the syncytial blastoderm is contained within its own little territory of cytoskeletal proteins. When the nuclei reach the periphery of the egg during the 10th cleavage stage or so, maybe the 11th, each nucleus becomes surrounded by microtubules and microfilaments. The nuclei and their associated cytoplasmic islands are called energids. Following division cycle 13, the cell membrane, which has covered the egg, folds inward between the nuclei, eventually partitioning off each energid into a single cell. This process creates the cellular blastoderm, in which all the cells are arranged in a single layered jacket around the yolky core of the egg. This is why this kind of cleavage is called superficial cleavage, because once the cleavage really starts to happen, it doesn't really happen as cleavage of cells until those cells form at the periphery of the ovum. If we look at how this happens, looking in panel C now, we see that the nuclei have come, diagrammatically, have come up to the area of the membrane of the ovum. Microtubules extend down around the nuclei, elongate that component of the cytoplasm and the membranes of the ovum. Actin forms at the bottom. You can see the actin filaments in cross-section there. And there's a pinching off then of that membrane from the ovum and a surrounding of the nucleus and the various components of the nucleus, the centrioles, microtubules, mitochondria, and so forth, to make that now a complete cell. And so that's really how the cell formation initially takes place, and it's not until about the 13th cell division that you get individual cells actually forming. Prior to that, it's simply a division of the nuclei as a component part of these energids. Let's now look at a schematic representation of gastrulation in the Drosophila. If we look at panel A, the general body plan of Drosophila is the same in the embryo, the larvae, and the adult, the general body plan. Some differences, obviously, but basically pretty much the same. And 3 of these segments form the thorax, while another 8 segments form the abdomen. If we look at this diagrammatically, this schematic representation of gastrulation shows that the anterior is to the left, dorsal is facing upward. In A and B, the surface and cutaway views show the fates of the tissues immediately prior to gastrulation. And you can see that in the anterior component, there's endoderm in yellow, the mesoderm is at the ventral aspect, red, and internal ectoderm, light blue. The amnioserosa, which is actually an extra embryonic membrane in a sense, it really forms a covering of the embryo, it's extra embryonic. And then there are pole cells, the germ cells, which will give rise to the sperm and the egg. As gastrulation proceeds, B, this is still pre-gastrulation, you start to get an invagination of the ventral aspect of the embryo, and going down to C, you can see that invagination forms initially a trough and then eventually a tube of mesoderm that will extend on the inner part of the body. By D, the germ cells have migrated ventrally and are now going deeper into the body of the developing embryo. And by E, you can see the nervous system has developed. The mesoderm in red has most inner part of the body, and also along with the endoderm. You can see that the endoderm on the outside has moved inward again to form parts of the gut and some of the other structures, inner part of the mouth, that are going to be components of the digestive system. If we look at this in a scanning electron microscope mode, we can see that gastrulation in Drosophila is pretty much, in many ways, like it is in some of the vertebrates, as far as some of the mechanisms involved. Different layers are different in the Drosophila compared to vertebrates, but nevertheless you get a ventral furrow formed, which is similar in a sense to the formation of the neural tube, which starts as a neural trough, a neural furrow, in frog embryos, for example. But here in the Drosophila, if we look at the gastrulation, the anterior of each gastrulating embryo, in this case now, points upward. That's sort of a standard way you look at Drosophila. The ventral furrow begins to form as the cells flanking the ventral midline invaginate. Looking at panel B, you can see that here at the anterior end is the prosencephalon. That's going to form the head. There's an invagination again of the gut. That's going to be the mouth. There's a area there that's called the cephalic furrow, which will separate the thorax from the head regions. And then the ventral furrow now in this case is starting to close, or has pretty much closed. You can still see a remnant of where the ventral furrow is. Going at a little more advanced stage, again, and looking at the dorsal aspect, you can see the head region here. The cephalic furrow is right down below here. You can see the pole cells in the dorsal aspect that are migrating dorsally from their former region. There's also a midgut invagination at that point. Here we are looking at the embryos in a side view, and you can now see the head region, which has mandible, maxillary, and labial components, the MA, MX, and LB, labial. So these are going to be basically mouth parts that are going to form there. You can see the formation of where the head will be then in this embryo is indicated in the diagram. You can see thorax components. There are 3 thoracic segments we mentioned before, T1, T2, and T3. And then there are abdominal segments that will actually end up 8 formed in total, 8 abdominal segments. But looking at this particular diagram right here where the arrow is indicating, the abdomen is sort of coiled around. A1 and A2 are going around here, and then the other up through A8 around the ventral aspect of the embryo. And now, going here, you can see it's further developed, and you can see now that these segments are pretty well defined. The mandible, maxillary, and labial portions of the mouth parts are present, and you can see other areas, the procephalic region, the optic regions, and the thorax forming. At the bottom, you can see now actually the beginning of a newly hatched first instar larvae. And so this is showing you gastrulation taking place in Drosophila. Here we see axis formation in Drosophila. We have the anterior portion of the embryo and the adult pointing upwardly. There's the head region, cephalic region. We have the thorax region indicated by the three segments T1, T2, and T3, which are called the prothorax, mesothorax, and metathorax. And then we have the abdominal segments A1 through A8. So there are a total of 11 thoracic and abdominal segments. The dorsal aspect is to the left now, as you can see in the embryo and in the adult. The prothorax in the embryo gives rise to T1 in the adult, which has the one leg. It has the anterior leg of this insect. The mesothorax has two appendages on each side, four total, two appendages on each side. It has the wing and the leg, and metathorax T3 has the posterior leg. So you have three pairs of legs and a pair of wings on T2. T1, T3 each have a pair of legs. If you talk about the anterior-posterior axis of Drosophila, as we mentioned earlier, it's really specified during oogenesis. This is put in place by the mother when the egg is being developed. The anterior-posterior axis then is visualized here. In A, the oocyte moves into the posterior region of the egg chamber, while the nurse cells fill the anterior portion. The oocyte nucleus moves toward the terminal follicle cells and synthesizes a protein called Gherkin protein, and that's the protein that's indicated in the green inset there on the right. And the terminal follicle cells express torpedo, which is another protein, and when there is an interaction between Gherkin and torpedo, when Gherkin binds to torpedo, the terminal follicle cells differentiate into posterior follicle cells and synthesize a molecule that activates protein kinase A in the egg. When protein kinase A is activated, this orients the microtubules such that the growing plus ends are posterior. If we look then in D, the bicoid mRNA binds to dynein, a minus-directed motor protein associated with the non-growing end of the microtubules, and the dynein moves the bicoid mRNA to the anterior end of the egg. Looking at E now, the nucleus, with its associated Gherkin protein, migrates along the microtubules to the dorsal anterior region of the oocyte and induces the adjacent follicle cells to become the dorsal follicle cells. Anterior-posterior axis is being specified, as well as superior and inferior axes too. And if we look at the photomicrograph of bicoid mRNA, stained black with this particular antibody, passing from the nurse cells and localizing to the anterior end of the oocyte during oogenesis. Keeping in mind that all of this is happening during oogenesis and prior to fertilization or zygote formation. Thus, the axis specification in Drosophila takes place early in development, totally under control of the maternal genes. Let's now look at the dorsal ventral axis. We started to get a little hint of a dorsal axis when the nucleus associated with the Gherkin in panel E here went toward the dorsal aspect of the embryo. But as we continue the dorsal ventral axis specification, this actually begins about 90 minutes after fertilization. But again, although it begins after fertilization, it is directed by the maternal RNA that's placed in the egg by the mother. The dorsal protein, as it's called, is a morphogen that is synthesized from the maternal message in the ovum, beginning, as we mentioned, about 90 minutes after fertilization. Although dorsal protein is present throughout the syncytial blastoderm of the Drosophila embryo, it is translocated to nuclei only in the ventral part of the embryo. It seems like a paradox, but it's because in Drosophila, mutant genes are named for what they fail to do. And so that's why we're calling a dorsal protein that actually turns something into a ventral part of the embryo. The dorsal protein does not enter the ventral nuclei. Genes that specify ventral cell types are not transcribed. And dorsal cell types are not repressed, so all of the cells become dorsal cells. Even though the gene is not functioning correctly, it promotes the dorsal formation, which is not really what it's supposed to do in the embryo. So, as I said, it seems like a misnomer, but genes in Drosophila are usually named for the mutant phenotype that they create. If we look at the specification of the cell fate then by gradient of dorsal protein, the largest amount of dorsal protein is actually should be in the ventral part of the embryo. And if that's the case, which is true in wild-type embryos, that will give rise to the mesoderm. And as you go up toward the dorsal part of the embryo, there's almost no dorsal protein present, and those cells then will actually become the dorsal part of the embryo. Looking at some mutants down below here, B, C, and D, if we look at B, this is a wild-type embryo where the dorsal protein has become incorporated into the cells in the ventral part of the embryo. This is a cross-section now, and so this embryo is going to develop normally. If we look at C now, this is a dorsalized mutant. So there's no dorsal protein in any nucleus, and because there's no dorsal protein in any nucleus, there are no ventral structures represented. In other words, there's no mesoderm. And if we look at transverse section D, this is the opposite, where all of the cells contain dorsal protein, and so all of those cells are ventralized. They become basically all mesoderm. So this kind of a gradient with dorsal protein, and there are other proteins that are involved as well, but this is a major protein which will cause superior, inferior gradients that will lead to the orientation of the embryo. If we look at the effects of the dorsal protein in this gradient, this shows the gastrulation now in Drosophila, and we have stained particular protein that will form in cells that have been ventralized by the dorsal protein. Those cells are indicated by the brown reaction product in the ventral aspect of the embryo here. Those cells have been treated with dorsal protein, and so those ventral cells have turned into mesoderm, and they're going to invaginate to form the mesoderm of the embryo. So this is really a form of gastrulation similar. This is called invagination, but it's similar to what's happening that you learned about in the amphibian embryo, where the cells that are going to form the mesoderm will involute inside of the embryo to form the mesodermal structures. And there are many mesodermal structures then that will form in these Drosophila embryos. I'd now like to move to segmentation and the anterior-posterior body plan in a little more detail. We talked about it somewhat when we talked about the polarization in the egg, but basically genetic screens were used to identify a hierarchy of genes that establish anterior-posterior polarity and divide the embryo into a specific number of segments, each with a different identity. This hierarchy is initiated by maternal effect genes that produce messenger RNAs. And here we see maternal effect genes indicated in the diagrammatic representation in this slide. The first such zygotic genes to be expressed are called gap genes, but the very first genes to be expressed are those that are maternal effect genes that produce the messenger RNAs localized in different regions of the egg. These mRNAs encode transcriptional and translational regulatory proteins that diffuse through the syncytial blastoderm and activate or repress the expression of certain zygotic genes. The first such zygotic gene, in other words, a gene that's produced from the zygote rather than just from the maternal genes, is called the gap gene. Here you see gap genes, and there are more than one gap gene. There are more than one kind. These genes are expressed in certain broad segments, partially overlapping domains. The gap genes encode transcription factors, and gene proteins regulate transcription factor of pair rule genes. And so the cytoplasmic polarity from the neural effects produce transcription factors which affect gap genes. The gap genes, again, factors which regulate pair rule genes as well as homeotic genes, and the pair rule genes then affect segment polarity genes as well as homeotic genes. So there's really a cascade of effects that lead to the anterior-posterior body plan. Together, the actions of these genes define the spatial domains of the homeotic genes that define the identities of each of the segments. In this way, periodicity is generated from non-periodicity, and each segment is given a unique identity. The maternal effect genes and the product of certain genes can be seen in the images on the right through in situ hybridization. Let's look first at the maternal effect genes indicated in panel B. The anterior axis is specified by the gradient of bicoid protein, yellow through red, yellow being the highest concentration. If we look at C, the gap gene protein expression is there, along with overlap. The domain of the hunchback protein is orange, and the domain of the Krupel protein, green. These overlap to form a region containing both transcription factors, which is indicated in yellow. And D has a rather unusual localization in the developing embryo. Here we see products of the pair rule gene. This happens to be the Fushi-Terazu pair rule gene. It forms 7 bands across the blastoderm of the embryo, and these bands are involved in the regulation also of the anterior-posterior pattern formation in the developing embryo. And the final one illustrated here are products of the segment polarity gene engrailed, that's E, seen here at the extended germ band stage. And so you can see that there's a localization of these various genes in terms of their products. The RNA products where in situ hybridization is formed, as well as protein products. And so clearly the segmentation is a complex set of interacting genes, beginning with the maternal effect genes that produce transcription factors and extending to a cascade of zygotic genes, resulting in segmentation. We have now covered the basic aspects of early embryonic development in Drosophila. Clearly, one of the greatest values of this animal model in modern cellular, molecular, and developmental biology research has been the tremendous collection of genetic information available, including a completely sequenced genome and a huge collection of mutant genes that affect so many biological processes and molecular pathways. Also, many of these genes are relevant to those in higher vertebrate organisms, including humans. Drosophila is indeed an amazing and valuable metazoan species for studies in developmental biology.